Bismillahirrahmanirrahim. Assalamualaikum and good day everyone. Okay, so this is the recording for our next chapter, which is the chapter 3. And in this chapter 3, we're going to learn anything and everything about the bacteria. This is the mind map that I have made. So this is to show that what we are going to learn in this chapter about bacteria. Okay, so the first one, we're going to have a look at the nomenclature or the naming system on how to write down and how to know whether this is the bacteria or not the bacteria. And the second one is on the taxonomy. So for the taxonomy level, we're going to have a look at the morphological characteristics, the physiological and metabolical characteristics, and also the molecular analysis or molecular characteristics. Then next, we're going to learn about the bacterial isolation on why we want to isolate bacteria. And during the bacterial isolation, the aseptic technique is very important to achieve the pure colonies. Next one is the bacterial identification. So once we have successfully isolated the bacteria, then it is time for us to identify. So for bacterial identification, there are two most basic and common method that we're going to use and we're going to learn. The first one is on the colony morphology and second one is on the gram staining process. Next, on the nutritional needs. So we're going to see whether the bacteria are autotrophs and also the heterotrophs. The next one is on the microbial growth. Okay, so for microbial growth, we're going to learn on the culture media. So what is the media responsible to aid the growth of the bacteria? Then we're going to have a look at the environmental factors that contribute to the growth of the microbe or the bacteria, and also on the determination numbers of the bacteria. Next one, we're going to learn about the epidemiology, which is the study about the disease caused by the bacteria. And the last one is just a brief on the antibacterial properties by the antibacterial agent. The first one, I'm going to learn about the bacterial taxonomy and also the nomenclature. Okay, so I believe that you are by now familiar with naming or the nomenclature of the bacteria. Okay, so for the bacteria, we have the genus and we have the species name. So for the family names for the bacteria, it always ends with the Aciae. Yeah, so for the genus name of the bacteria, it has to be capitalized. Yeah, and also when we type in the laptop or in the desktop, yeah, so the genus and species has to be italicized like this, the Staphylococcus aureus. But then when we write it down using the pen or using the pencils, so the genus and species can be underlined just like in the slide. Okay, so for the species name, I'm sure that you have seen this, the SP and also the SPP. So we use the SP and also the SPP when the species is not identified. When we don't know what the species is, we only know the genus. So for example, Staphylococcus. Staphylococcus is the genus, but we don't know the species of the Staphylococcus. Yeah, so we can use the SP to refer that as a singular or one species of the Staphylococcus, or we can use the SPP to denote that it is a plural, or it means that several species of the Staphylococcus bacteria. Abbreviations, okay, so you are familiar with this. So for example, Staphylococcus aureus, so this is the full name. So if you want to write the abbreviation, so the genus name, we take the first letter of the genus name and then put a dot and then we write down the full name of the species name. Okay, so for the taxonomy, so there are major characteristics that we can use to do the taxonomy. Okay, so we can have a look at the morphological characteristics. We can look at the physiological and metabolic characteristics, on the ecological characteristics, and also the molecular characteristics. These are the characteristics or the criteria or the parameters for the morphological characteristics. So when we talk about the morphology, so it means that the shape or the outer layer or something that we can see. So for example, cell shape. So the shape of the bacteria, it can differentiate the bacteria, based on the shape. Based on the color as well, we can differentiate the bacteria. Based on the staining behavior, okay, whether they are the gram positive or the gram negative, and also the way they move, how they move, what do they use to move, then that data or that information can be used to characterize the bacteria. Next one, we can use the physiological, metabolic, and also the ecological characteristics or the parameters. Yeah. So, for example, the carbon and nitrogen sources. So, whether the bacteria they use the carbon and nitrogen sources to survive, or from where do they get the carbon and nitrogen sources? So, those are the categories that we can use to identify and also to characterize the bacteria. What else? Okay, so for example, the growth temperature, optimum, and also the range. So the temperature range that the bacteria use to grow, then that can be used to characterize the bacteria, or the pH, or the salt requirements, or the environmental or ecological characteristics that can be used to differentiate the bacteria. Next one is on the molecular characteristics. So previously, okay, historically, usually we use the percentage of the GC or the guanine and cytosine. But right now, due to the modernization, we can use the hybridization technique or analysis, and also the gene characterization in order for us to characterize the bacteria based on the molecular level. Next one is on the bacterial isolation. Okay. So the reason why we isolate the bacteria is because we want to have a single colony. Okay, and when the bacteria they grow in groups, then it is called the colonies. Okay, so next time, anytime you hear about people saying about the colonies, then it refers to the bacteria that grow in groups. Okay, so it is not a single colony, it is not a pure colony, but simply the colonies. It means the bacteria that grow in groups, and the bacteria they must grow on an agar, or on a growth media. Okay, so growth media or agar or culture media, so you will hear quite a lot terminologies on the growth media or the agar, but actually the growth media and agar and culture media, it is something that aids or something that help the growth of the bacteria. Okay, so I'm sure that you have done this in the lab. So in order for us to do the isolation of the bacteria, we use the strict plate method, and the strict plate method is on the zigzag method. Yeah. So again, the bacteria isolation, the reason why we do the bacteria isolation is to separate the colonies to get a single colony. So when we do the bacterial isolation, aseptic technique is very, very important. Okay, so aseptic technique is a technique or the procedure um, used by the people who want to do that procedure, to prevent the spread of infection of the bacteria. So in this case, it's the bacteria. So we don't want to contaminate the area that we work with, and we don't want to contaminate the sample that we are working with. Yeah, so this diagram or this figure, it shows on what we should do to achieve that aseptic technique. So once we have successfully isolated the bacteria, then it is time for us to identify the bacteria. Now when we have that single colony, then it is easier for us to identify the bacteria because we know that there is only one bacteria. So for us to identify the bacteria, okay, so there are a few ways, but in this lecture, we're going to learn about the two most basic method or ways to identify the bacteria. The first one is through the colony morphology, and the second one is on the gram staining. Okay, so colony morphology. So again, the morphology, the terminology comes out again. So morphology, it means that something that we can see, something that we can look on the outer layer of that cell or the bacteria. Okay, so for example, the shape, the margin, the optical properties, the pigment, yeah, the color, elevation, and also the texture. So these are the examples of the colony shape. So whenever you grow your colonies on an agar, and then you can have a look at the shape, whether it is round or filamentous or rhizoid or irregular. So this is the shape. So to see the colony shape, we can just see from the top view of that agar plate. Next one, we can see, we can determine we can identify the bacteria based on the colony margin. Okay, so whether it is entire smooth or round radiating or irregular, okay, or ciliate. So this is a reference for you, for us, when we want to identify the bacteria based on the colony margin. Next one is on the optical properties, okay, whether we can see through the colony on the agar. So if we can't see through the colony, okay, from the top view, then means that it is an opaque. But if we can see through just barely, then it is translucent. And if we can see through all the way from the top view to the bottom of the agar plate, then it is transparent. Also, we have another part where it is glossy. It means that it is shiny. Colony pigment, yeah, pigment is on the color. So we can see that, and I know that you have experiences as well in the lab. And so some of you have successfully grown bacteria from different sources and with different colors as well. Okay, some of them are yellow, some of them are red, some of them orange. Okay, so these different colony pigments, it can be categorized into different type of bacteria. Okay, next one is the colony elevation. Okay, so colony elevation, for us to identify the bacteria based on the colony elevation, we have to look at the agar plate from the side view. Okay, then we can see whether the colony elevation is raised or flat or drop-like and so forth. The next one is on the colony texture. So in order for us to identify based on this colony texture, we have to touch the colony. So remember, when you want to touch the colony, aseptic technique is very critical. So because we don't want to contaminate our plate, and we also don't want to contaminate the area of our lab, okay, with the sample that we have in the agar. So for the texture, when we touch it, we know that whether it is soft or granular or tenacious or mucoid. Okay, so that is the first common and basic way on how to identify the bacteria through the colony morphology. The second one is on the gram staining. So gram staining is to gram staining is a process in order for us to categorize bacteria or cells based on whether they are the gram positive or the gram negative. Okay, because gram positive and the gram negative, this is the two large groups or the two main groups of the bacteria, and it has a series of reagents or stains that we need to use. Okay, and the result is, if the cells are stained with purple, then it means that it is a gram positive bacteria. But if the cells are stained with pink color, then it means that it is a gram negative bacteria. So this is a figure showing the composition of the bacteria, and on the left hand side is the gram positive and the right hand side is the gram negative. Okay, so what is the major difference between the composition of the gram positive and also the composition of the gram negative? So one thing that we can surely see, obviously, is on the peptidoglycan layer, okay, which is the one colored in purple. So for gram positive layer, the peptidoglycan layer is very thick, and however for the gram-negative layer, the peptidoglycan layer is very thin. Okay, so as mentioned just now, gram-positive, it has a very thick peptidoglycan layer because it serves as a protection to the cell of gram-positive bacteria. On, on the other hand, gram-negative, it has a thinner, much thin peptidoglycan layer than the gram-positive. Okay, so this is the process for the gram staining, and we will continue